Assalamu alaikum. Welcome back to the Guyton Academy. This is the third practical from the series of physiology blood practicals. This is the VIVA and OSPE question guide. This basically this all these videos are designed for the preparation of OSPE and VIVA questions that may be the part of observed and unobserved OSPE in the physiology practicals of blood. So this is the practical how to use microscope. This practical is also a part of anatomy also. So the first question that may become an exam is what is microscope? A microscope is the instrument which produces an enlarged image of a small object, and it reveals the details which are too small to see with our eyes. The next question would be what is the compound microscope? A compound microscope is defined as a microscope with high resolution and uses two sets of lenses, providing a two-dimensional image of the object. This is the picture of a compound microscope. I am not going to discuss all the labelings. You just pause the video and you can learn the labeling of the microscope. That will be helpful in your exam. If the labeling will be coming in your exam, maybe this may be the part of unobserved OSPE. Okay, so I'm not going to discuss it. It will waste a lot of time because you have to memorize it, the parts of the microscope. Now, what is the function or use of microscope basically? Maybe in some questions you will feel that they are repeated, but because the way of asking is different by different teachers, so we will try to cover maximum questions from this topic, right? So what is the function? The function of the microscope is to magnify the objects that are too small to be seen with our naked eyes, and it helps in diagnosis of the many diseases by in the laboratory. Now, what is the difference between monocular and binocular microscope? Basically, the two broad categories of microscopes are monocular and binocular. So, monocular microscope has one eyepiece and one eye tube. If you see this picture, this is the eyepiece and this is the tube. So, in the case of monocular microscope, we have one eyepiece and one eye tube. Magnify sample up to 1000 times. It can magnify sample up to 1000 times. Why? I will discuss in the later stages. It is used to observe the samples on the glass slides like animals, small animals, plants, and cells. What is binocular microscope? In the case of binocular microscope, we have two tubes and two eyepieces. This is the binocular microscope. It has two eyepieces and two tubes. Magnifying samples up to 1000 times, because in the case of simple monocular and simple binocular microscope, the magnification power is 1000 times. More comfortable to examine a specimen. Binocular has because two eyepieces, so we can use both, we can open both the eyes, so it is very comfortable to examine with the help of binocular microscope. It is used in educational, research, and commercial settings. However, monocular microscope is mostly used in the schools for a small work, you can say. What are different types of microscopes basically? So there are about 16 types of microscope. However, the most famous microscopes are simple monocular, simple binocular, compound microscope, compound binocular, fluorescence microscope, and electron microscopes. Define magnification and its unit. Magnification is defined as the enlargement of the object produced by the microscope, and its unit of measurement is diopters. Many times this is asked in your unobserved OSPE, what is diopters? Now, what are the different parts of the body of compound microscope? The compound microscope consisting of eyepiece, objective lenses. Basically, eyepiece is also a lens, okay? Eyepiece, objective lenses, stage, adjustment knob, condenser, diaphragm, illuminator unit or light source. What are the different parts or components of magnifying unit of a microscope? In the previous slide, we asked about the body, complete microscope. In this slide, we are just talking about the magnifying unit of the microscope. The magnifying unit enlarges the image of the specimen, and it consisting of two main parts, eyepiece and objective lenses. In the compound microscope, eyepiece has two lenses. Binocular microscope has two eyepieces. What does it mean? It means that in a compound microscope we have two eyepiece, right? This is called ocular lens. In each eyepiece we have two lenses, right? Each eyepiece has two lenses, and binocular, because it is binocular, so it has two eyepieces. So in total it has four lenses in two eyepiece. And objective lens, these are the objective lenses. We will discuss in detail in coming slides. So basically, magnifying unit of the microscope is eyepiece and objectives. Some more details about the magnifying unit of the microscope. Objectives. Basically, objective is and eyepiece are the lenses. ठीक? This is not the objective of your research. This is the objective, means the objective lens. So this lens is closest to the specimen. This is the slide where we place this is the place where we place the slides, and on the slide we have a specimen, and the nearest lens here is the objective lens. So these are the objective lens which are connected here with the nose piece. So the lens is closest to the specimen. It produces primary magnification image. It comes in various magnifications. It is asked in your exams what are the different magnifications of the objective lens. So it can be 4 times, 10x, 20x and 100x. Similarly, eyepiece is also a lens. It further magnifies the image produced by the objectives. First of all, the image is produced by the objective lens, and then it is further magnified by the help of eyepiece. Eyepieces typically have a magnification of 10x. This one. How to calculate total magnification of microscope? By multiplying the magnification of the objective lens by magnification of the eyepiece, we get the total magnification of a microscope. For example, a 10x objective used with a 10x eyepiece would provide a total magnification of 100x. What is the stage in the microscope? The stage in a microscope is a flat platform where the specimen, usually on the glass slide, is placed for the observation. This is the stage where we place the slide and a specimen is placed on the slide. So stage is a flat platform. In the center of the stage there is a circular hole for the light from the below to pass through, so that we can see the specimen. This is the light source. So when it passes through from the lower portion, it will come to the specimen and we can see it with the help of, first of all, the objective and then the eyepiece. Which objective lens is typically used for initial focusing of the specimen? So we have a 4x objective lens, which is also known as scanning objective, is used for the initial focusing. Fine and coarse adjustment knob. There are two knobs. This is the fine adjustment knob and this is the coarse adjustment knob. So the coarse adjustment knob is used for the initial focusing or rough focusing of the specimen by moving the stage up and down quickly. Actually, it will just adjust the position so that we can see the specimen. However, Just after the coarse adjustment, there is fine adjustment. After the coarse adjustment, fine adjustment knob is used for fine focusing of a specimen by moving the stage. In both the cases, we are moving the stage, but for the coarse adjustment, there is rough placement of the specimen so that we can see, at least see the objective, and to clearly see, we use the fine adjustment. What is working distance? This is the slide and this is the objective lens. So the distance between the slide or specimen and the objective lens, this is called the working distance. Working distance is the term used for the distance between the objective lens and the specimen. What is the use of illumination unit of a microscope? It is responsible for providing the light that illuminate in the specimen. This is the illumination unit, right? Or you can say light source. So the components of the illumination unit of a microscope are light source. This is the bulb, condenser lens. There is a condenser lens, iris diaphragm. This is the iris diaphragm, and then there are some filters. So the components of the illumination unit are light source, condenser lens, iris diaphragm, and filters. What is the focus adjusted? How is the focus adjusted when using a microscope? The focus is adjusted using both coarse adjustment and fine adjustment. How can the field of view be adjusted? The field of view can be adjusted by moving the slide forward or backward using the stage knobs. What is the first step when using a microscope to examine a slide? The slide should be placed on the stage and secured with slide holder. How does the objective lens of a microscope differ from ocular lens? Objective lens magnifies the specimen directly because this is close to the specimen. And then ocular lens, this is the ocular lens. Ocular lens further magnifies the image created by the objective. What is the magnification power of typical high power objective lens? It is 100. Why do microscope need a light source? Because light provide illumination for the specimen. How does a microscope achieve higher magnification? By using a combination of objective and ocular lenses. What are the different types of objective lenses used in the microscopy? We can have a scanning, low power, that is 4x. Low power can be 10x, and then 20x, and then 100x, or oil immersion. 100x is also known as oil immersion lens. So basically these are the 4 different types of lenses. What is the purpose of using a cover slip over the specimen on a slide? Cover slip protect the specimen, it prevent from evaporation, and it create a more even surface for focusing. What is the difference between light microscope and an electron microscope? The light microscope use visible light to magnify, while electron microscope use electrons. Now this is the same question. When is oil immersion used and why? Or why it is important to use immersion oil with 100x objective lens? Oil immersion is used with 100x objective lens to improve the resolution and clarity of the image. It helps to reduce the refraction of the light that occurs when the light passes from the slide to the objective lens. What is the role of the stage in microscope? It holds the slide in place. What is the ideal surface for placing a microscope? Microscope should be placed on a firm, uniform, and smooth surface to prevent the vibrations that can blur the image. What is the use of condenser iris? What factors should be considered when adjusting the condenser iris? Condenser iris control the angle of the beam of light. focused onto the specimen. With the help of here, we have the angle of beam of light, which can be controlled by condenser iris. Basically, the type of a specimen being examined should be considered. What factors should be considered? Type of a specimen should be considered to adjust the condenser iris, because the stained and mounted preparations generally require a wider aperture. Means when we have a stain, a slide or a specimen, it need wider aperture, that is more light. Unstained objective may benefit from the reduced aperture, that is low light. What is the function of revolving nose piece on a microscope? This is a nose piece. Allow for easy switching between objective lenses. Which microscope objective lens must be used to start with when viewing a specimen and why? Means initially हमें कौन सा use करना चाहिए? We always start with the lowest magnification to locate the specimen and avoid damaging it, because its size is also small, so it will not touch the slide. So first of all, we will try to use this, and then slowly and gradually we will change our objective lenses. What is the numerical aperture of a microscope lens? A measure of microscope lens ability to gather light, that is the numerical aperture. Higher numerical aperture objectives collect more light, produce a brighter and better quality of the image. How does microscope resolution differ from its magnification? Or you can say, what is the difference between resolution and magnification of a microscope? Resolution is the ability to distinguish between two closely spaced points, while magnification is the enlargement of an object. Resolution gives you the high quality image, magnification gives you the large size image. What is the importance of proper illumination in microscopy? Ensure clear and detailed image. How does the working distance change with different objective lenses? Higher magnification objectives have shorter working distance, because the higher magnification objective have more larger size, so the working distance will be reduced. What is the difference between bright field and dark field microscopy? Bright field microscopy illuminate the specimen directly. Dark field microscopy illuminate the surrounding background. How do phase contrast microscope differ from traditional light microscope? Phase contrast microscope use phase shifts to enhance the contrast of unstained specimen. What are the advantages of using a binocular microscope over a monocular microscope? Actually, the binocular microscope reduces the eye strain and provide a more comfortable viewing experience because both the eyes are open. What is the purpose of light intensity control in microscope? To adjust the brightness of the illumination. Light intensity adjusts the brightness of the illumination, that is how much light should be provided to the specimen. What is the refractive index of immersion oil and why it is important in microscopy? Refractive index matches the refractive index of the glass. Refractive index of immersion oil matches the refractive index of the glass, reduce the light refraction and improve the resolution. What type of microscopy techniques commonly utilize immersion lenses? Bright field, dark field and phase contrast use immersion lenses. How do immersion lenses improve the depth of field in microscopic imaging? By reducing the light scattering and improving image clarity. Why it is important to match the refractive index of immersion oil to the lens and slides? To minimize the light refraction and improve image resolution. Thank you. I hope this will be very beneficial for you. As a token of appreciation for my hard work and dedication, I would like you to subscribe my channel, like the video, and give some comments about this video. I hope this will be very helpful for you. Thank you so much.